All right, we are going to be on pages 14 and 15 for Lab 6, looking at the nervous system. We're going to start looking at the nerve cell, which we call a neuron, and then we're going to go into the brain models and then end up with the sheep brain dissection, looking at parts of the brain as it relates to the sheep. So if you look through the lab packet, you'll see that there's some things that have an asterisk. So be sure that you're identifying those like on a microscope slide versus a sheep brain. So when we get to the actual brain parts, if there's an asterisk by it, it means you need to know not only on a model or a diagram, but also on the sheep brain itself. So looking at our first set of objectives here, looking at the parts of a multipolar neuron, we have two different neuron models here. The first one, the one on top, you can see they have some common characteristics. There's a enlarged end with these projections on it, and then we have kind of a long narrow piece, and then we have the end of the neuron that has those little extensions on it, which are the axon terminals. So starting from the enlarged end, this is the cell body, this large portion. And looking at the cell body, you're going to find a nucleus, which is this white space here. So that has the nuclear membrane, which we looked at when we studied the cell. Here's another one. Here's the nucleus with that white nuclear membrane. So the larger circle or space is the nucleus. And then inside the nucleus are these smaller organelles. There might be one or two, and that's a nucleolus. More than one, we call a nucleoli. So nucleolus or nucleoli. So just be sure you don't think of that as the nucleus. The larger rounded space outside of that is the nucleus, and this is the nucleoli. So nucleus, nucleoli. And then these extensions coming off of the cell body, the white ones, ignore the brown ones here. These are axon terminals from other axons or other neurons. So just the white extensions here, those are the dendrites. So that's the receiving end of our neuron. So information is coming into the nerve cell from the dendrites to the cell body to be processed, and then if that information accumulates, we have enough ion flow, enough sodium and potassium movement across the membrane to get what we call an action potential, which is the impulse that's carried down the neuron. Then that leads us to the axon. So this elongated, thin portion carrying information away from the cell body is the axon. So both of these here, two different, neuron cell models, but they have the same parts, just different representations. So the long, thin portion is the neuron. And what covers the neuron, the axon, what covers the axon is this fatty coating. Not every neuron in the body has an axon with a fatty myelin covering, but a lot of them do, especially the ones that come through our spinal nerves. They have this fatty covering. This is myelin. So these white layers are myelin. And if I look at this neuron here, the green covering is actually the plasma membrane, because the plasma membrane covers the entire cell. So not the green covering, but the yellow covering down here is myelin. So the white layers in this model are myelin, and the yellow here is myelin. So the actual axon in this model down below is the gray part. That's what's inside there, underneath the myelin. So the myelin provides kind of a protective, insulated covering over the axon. And then we have, the axon underneath that. And there's a cell that makes the myelin. It kind of looks like a spidery structure here in this model. This is called a Schwann cell. So in this model here, you can see this little orange cell. That's the Schwann cell, secreting the yellow myelin over the gray axon. So in that bottom. And then again, the plasma membrane is green and it covers the whole thing. So in this top model here, that little cell right there, that's the Schwann cell. And it's secreting myelin below it. So these are special helper cells in the nervous system that have a specific function. And Schwann cells, their job is to make myelin in the peripheral nervous system. So it deals with axons in nerves, those that are extending off the brain and spinal cord. So that's a Schwann cell. And then there's these spots, there's bare spots on the neurons. So this little indentation right here is where there's no myelin, And we find voltage-gated sodium and potassium channels there because that's where the impulse is going to jump, is from node to node to node. And that node is called a node of Ranvier. We talked about that in lecture also. But that's the node of Ranvier. And in this model, it's an orange spot. It's like a little disc they put on the axon called the node of Ranvier. And it looks kind of like a lifesaver wrapped around the axon. And it's not really the best representation of a node because a node is a bare spot on the membrane. It's not a specific structure like it is with this little red or orange piece right here. It's a little more accurate in this model in that it's an indented kind of a thin bare spot on the membrane. And that impulse is going to jump from this node to the next node here. And it just speeds up transmission. Just like if you were running across a stream and there were rocks spread apart. So if you wanted to get across quickly, you'd jump from rock to rock to rock, you know, using the furthest ones apart as possible to get across the stream. So that's the node of Ranvier. Any questions on that? So we've got two different models. Be sure that you know these structures on both models. I like this green one here. It's because it shows the plasma membrane, because we have to remember all cells have a plasma membrane, have a nucleus, and cytoplasm. So regardless of their shape in the body, they're going to have those three structures in common. But we can see a nerve cell looks very different than like a skin cell, which is a flattened, you know, squamous cell. So then the tissue that we're looking at in this unit is nervous tissue. And remember, we looked at this in, you know, our second or third lab. I think it was the third lab. And nervous tissue has these large neurons. So what do you need to know in the nervous tissue? You need to know a neuron. And that's this large structure with the extensions coming off of it. You don't have to know the specific parts in the slide. Just know that when you look at it, what's the name of the tissue? Nervous tissue. And what's the cell? It's a neuron. So the larger cells. These smaller, darker structures in the background are supportive cells that we see in nervous tissue. And we'll talk about those more in lecture, like microglia, Schwann cells, oligodendrocytes, a couple of other supportive cells. And those are the ones that make up cancerous tumors in the nervous tissue. When someone says they have a brain tissue, it's not the neurons that start to grow out of control. It's these supportive cells that grow out of control and form tumors. And because their job is not to conduct impulses, those smaller supportive cells, they crowd out the functional portions of the nervous system, and that's what causes death eventually, is when these brain tumors get too large, they crowd out the neurons and the neurons can't function. Another structure to be familiar with is a nerve. A nerve is shown here, and a nerve is just a bundle of the axon portion of neurons. So the cell bodies are going to be found in the in bundles closer to the spinal cord or in the spinal cord, and then the axons extend away from that to make up nerves. So nerves are just bundles of axons outside the central nervous system. So it has a connective tissue sheath. They have a good blood supply. You can see there's an artery and a vein here. And then we have these bundles of myelinated axons. See the light blue covering over that surface? That's the myelin covering that axon. So bundles of axons is what makes up a nerve. So you have to know what that looks like in a tissue slide. So here's a tissue slide showing a nerve. And I can tell this is a nerve because I see all these small rounded structures, which are tiny little axons. And if I zoom in a little closer here, I gotta find my plus sign. Where's my plus sign? There it is. So if I look in closer here, so see the dark structure in the center here, right at the tip of the pointer? That dark structure is the actual axon, and what's the white stuff around it then? Myelin, yeah. So you can look and see that each one of these has an axon with a fatty covering on the outside. So this is just a bundle of axons, and a bundle is called a fascicle. So if I bundle up a bunch of axons together, put a connective tissue wrapping around it, we call that bundle a fascicle. And you'll notice that in looking at this picture here, This is one whole nerve here. So here's the outer covering of the nerve, and a nerve is made up of several fascicles, which are just bundles of axons. Think of them almost like, the axons like a spaghetti noodle, that's not cooked. You just bundle a bunch together. Those are all axons, put some myelin on the outside, making up a nerve. So several bundles of axons within a nerve. So that's some of the microscopic anatomy you're going to have to know for the next lab exam. And then we'll get into the gross anatomy, which is looking at models and the sheep brain. So if you take a look at the brain model that you have in front of you, or looking at this picture here, the brain is made up of a series of grooves and valleys. And the ridges, the enlarged swellings of the brain, those are called a gyrus. So if you put your finger just on one, it's called a gyrus. And then the valley in between the gyri, so the grooves in the brain, those are called a sulcus. More than one, we say sulci. More than one gyrus, we say gyri. So the brain is made up of gyri and sulci, and those are just provides extra surface area for these neurons to make connections throughout the brain. So a gyrus and a sulcus. The gyrus is the rise, the sulcus is the valley in that tissue. And the more grooves we have in the brain, the more advanced mental processing that can occur. So some people are born with developmental and chromosomal abnormalities that they have fewer gyri and sulci, and as a result, they're lower functioning. When you look at people that need total care, right, they're totally dependent on caregivers, they're in a wheelchair, they can't eat on their own, they're incontinent, they can't talk, they even have trouble swallowing. They have a lot fewer of these grooves because they're, developmentally the brain is not where it should be. So the more grooves, the higher the mental processing, because there's a greater surface area, there's that many more neurons for making connections and processing. So then if we look at the inside of the brain, put it in a sagittal view, so we cut this in half and just look at this view here. This is called the diencephalon. It's the central portion of the brain. So if you think of the brain, the cortex sitting on top of a central stalk, the inner portion here is the diencephalon. So this region here in the center of the brain, it's kind of like if you think of the brain like a walnut, and the shell of the walnut is the cortex, which is this wrinkled outer portion of the brain. That's the cortex. Then the nut of the walnut, the part that we eat, is in the center. And that's the diencephalon. And it has two lobes, you can see, so that's where it gets the name di, meaning two. And this is the thalamus here, which is the very central portion of our brain. And we'll talk about that. And then if we could extend from the thalamus, this diencephalon, this central nut of the brain, or if you think of the brain like a peach, the nut of the peach would be the diencephalon, which is made up of the thalamus and the hypothalamus. And there's other parts of it, but we don't hold you accountable for that. But the brain stem then is the stalk that the brain sits on. So there's three parts of the brain stem, three major parts that you need to know. We'll talk about those in detail, but for right now, just know them as the brain stem. And then coming off the back of the brain here is what's named the little brain, or little cerebrum, and that's the cerebellum. So it has these grooves in it. It's the cerebellum. It comes off the back of the brainstem. And then beyond that, so we have the cerebellum. So this is the brainstem that's shaded in green. And then once it kind of narrows and just becomes, at its thinnest point here, that's the spinal cord. And that's going to continue on down the spinal column through the vertebrae, right through the vertebral foramen, well protected. So spinal cord is where it's beige here and is at its most narrow point. But it's not, the spinal cord is not the same thickness throughout. It does have enlargements, like where the arms and the legs and the nerves come off to serve the arms and the legs, we do see some enlargements there. But looking at the brain model, where it's the thinnest point is the spinal cord. All right, and then if we zero in on the brain, there's coverings over the brain, but let's just look at this picture here. It's kind of neat. On the outside of the cerebellum, there's just these kind of grooved lines, but if I cut it in cross-section in a sagittal view, it almost has a cauliflower appearance, doesn't it? Or a tree look. Doesn't it kind of look like a tree? That's a really unique characteristic of the cerebellum when we cut it in cross-section, so you can identify how different that looks from the brainstem, which it kind of hangs off of on the backside. So the coverings of the brain that protect the brain are called meninges. And if you've heard of meningitis, that is a bacterial infection that is particularly dangerous for people that are living in close conditions. So we see bacterial meningitis to be a problem like in college dormitories, or in the military, where you have lots of people living together, in a close environment. So we recommend that young people get an immunization for meningococcal. Maybe you've heard of that. Maybe you've had that before recently. It just protects you from getting that bacterial meningitis, which is an inflammation of the meninges. And if you have inflammation of the meninges, that's a pretty serious, illness, because that's the coverings over the brain, and that can affect brain function if it's not controlled, and can increase pressure on the brain and do permanent damage. So it's a serious illness that we want to avoid if possible. So looking at the layers then, so this is, the skin, the bone, or the connective tissue covering, and then we have bone, then we have a basement membrane. But looking at this first layer, we have the dura mater. The dura mater is the toughest layer over the brain. So we can see there's kind of two layers here in this model. That's the dura mater. Both of those make up the dura mater. So it's almost like a thick canvas over the brain to protect it. And then we have a picture here on the sheet model, or I have a demo sheet brain that shows the dura mater, but it's a thick canvas-like covering over the brain. Durable, you've heard of that word before. Dura means tough or protective. And mater means mother. So dura mater means protective mother. Or tough mother. Sorry, it means tough mother. And then the next layer here, it has these little wisps coming off it. This is the arachnoid mater. And arachnoid means what? Spider. So it means spidery mother. And the reason why it's given that name is because of these little extensions that come off, and those actually are made up of cells. The dura mater is a combination of connective tissue and cells that secrete cerebral spinal fluid. So there's a space below the arachnoid mater before we get to the pia mater, this thin membrane that adheres tightly to the brain itself. This space here is called the subarachnoid space. Sub means below, so it's below the arachnoid mater. The subarachnoid space is where cerebral spinal fluid circulates, and that nourishes the brain. And the brain is actually floating in this cerebrospinal fluid, so it decreases the weight of the brain. So if someone falls backward and bangs their head really hard, that actually pushes the brain, that blow backward pushes the brain to the front, and people can end up with a really bad bruise on their forehead when they actually hit the back of their head, because the brain just shifted in its cavity. And that's also really bad for brain cells to be shifted around like that. So when people have a serious head injury, like a concussion or a traumatic brain injury, they actually shear some of the surface off of the brain, and that takes time for that inflammation to go down and that healing to occur. And how do we, if you sprain your ankle and you have inflammation of the ankle, what does the doctor tell you to do? Ice it, rest it, right? Rest is the biggest thing. Whenever we have an inflamed tissue, we rest it. Well, how do you rest your brain if you just had a concussion or a serious head blow? You could put some ice on it, yeah, but that might give you a headache. But how do we rest the brain?
Like, don't watch TV, use electronics.
Yeah, don't use electronics, don't look at bright lights. Keep the sound down, right? Keep the stimulation down. Don't study for things. Don't think about really difficult tasks, right? So if you or someone you know, or maybe your kids one day suffer a concussion, it's really important to take that seriously. Because I've had patients when I worked on pediatrics for a while, we had parents that, had kids that were in the hospital because of a serious concussion. And we tell them, low stimulation, no TV, no iPad, and they don't listen. Grandma and grandpa come in, they're bringing, magazines. They shouldn't even be reading, if they're in that early healing stage. I mean, we're talking about the first couple of days after a serious head injury. Keep the stimulation low. But a lot of times the parents don't listen to that, and the kids don't either. And then these kids have bad headaches, and they've got long-lasting problems. And we can really reduce the effect of concussion if we just listen and understand that those brain cells have been traumatized, and we need to rest them by quiet, low activity, hanging out, maybe playing a board game, kind of boring board game, not one that requires lots of concentration, right? And I speak from experience. My son had a serious head injury from a sledding accident at a field trip, and he hit his head on the icy hill and passed out, woke up, threw up all over the hill, all classic signs of a serious concussion. He cracked his skull. He had a brain bleed, so he had a traumatic brain injury. He was in critical care for one night and then went to pediatrics for 4 days. But knowing what I know, I kept the environment absolutely dark and quiet. And I sat in the corner on my Kindle in the dark, and I watched Breaking Bad over and over. I think I watched a couple of seasons. But the point is he slept, he was quiet, and he did nothing for 3 days. And he's perfect today. They were amazed that he had no lasting impacts from that serious injury. But people wanted to visit. Our pastor came by and wanted to come. I said, no, we really don't. And he forced his way in anyway and wanted to come and talk to him. So it was a little frustrating. I appreciate that he wanted to speak with him and stuff, but we had to really limit it and say, okay, people wanted to visit, and we said no. So it's hard to do that, but in the hospital, especially with the brain injury, people need to rest. So if you're ever told that, or someone tell them they gotta rest that brain, because they'll heal faster and they'll have fewer long-lasting effects by respecting that. So this last layer then is the pia mater, which is this thin membrane that goes down the grooves of the cortex. So this is that wrinkled portion of the brain. So this is a sulcus here, right? And this rise here is the gyrus. So the pia mater, pia means affectionate, Latin or Greek, I don't know which prefix there, but means affectionate mother. So that's because it's tightly adhered. So it's a very thin membrane. You'll see it when you dissect your sheep brain. You can get your needle probe underneath it and kind of tease it away. So you've got the dura mater, arachnoid mater, pia mater. So if we're looking at just specific layers here, you could point to this bottom layer here if you want to just kind of keep them right in a row. So the beige layer, the pia mater, dura mater, arachnoid mater, pia mater, in order from top to bottom. And then that subarachnoid space is where that cerebral spinal fluid is found. Okay, then we'll go to the nerves that come off the brain. There's a couple you have to know. There's 12 pairs of cranial nerves. So these are going to be lighter in appearance when we look at the brain because they're myelinated. They have that fatty insulating covering. So this is a cranial nerve here. Here's a cranial nerve. There's some other ones here that have been cut. Here's a cranial nerve. You don't have to know all these. The only ones we want you to know are the olfactory. This here is the olfactory bulb. You can see there are the swellings at the end. That's the olfactory bulb. And what is olfaction for? We say olfactory. What is it? Smell. Yeah. So these are for smell. So the olfactory bulb. And when we look at the sheep, we're going to see these olfactory bulbs are a lot larger. Because don't animals rely more on smell to navigate their environment and their world than we do? For sure. So this is a human brain, and this swelling is the olfactory bulb. And then this part here that's adhered to the brain, so we're inside the central nervous system, a bundle of axons inside the central nervous system is called a tract. Outside the central nervous system, we call it a nerve. But it's the same thing. Nerve or tract, same thing, bundle of myelinated axons. So because we're in the central nervous system, these these axons that make up this structure here is called an olfactory tract. So olfactory bulb, olfactory tract. Then we have the optic nerve, and these have been cut in this brain, but if we looked at this little stalk right here would extend out and end in an eyeball, at the back of an eyeball, but they've been cut in your sheep brain and also in this particular model. But the top of the X, can you see an X here kind of structure? So the top portion is the optic nerve that's been cut real close to the brain, but each of these would end in an eyeball. And then that information, so we've got an eyeball out here with let's say it was still intact, that optic nerve comes in, and then it crosses over to the other side of the brain. So behind this, where it comes over this crossing point here, this is the optic tract. So now it's part of the brain. It's in the central nervous system. So again, we change its name. It's still a bundle of axons carrying that light information to the brain, but we call it the optic tract. So the same thing on this side. We have an eyeball bringing things in through the optic nerve, crosses over, and at the bottom of the X there is the optic tract on that side. Where it crosses over, so right in the center of this X here, is what we call the optic chiasma. So the chiasma is the crossing point where that light information crosses over to the other side of the brain. So the top of the X is the optic nerve, the bottom of the X, optic tract, middle of the X, optic chiasma. And there's a nice picture in your lab packet there that kind of shows, you know, how that looks with the eyeballs attached. Or otherwise your textbook has a picture too. If you look up optic nerve and optic chiasma in your textbook. So those are the cranial nerves that you have to know. Then if we look at the grooves in the brain, kind of like we saw the sutures in the skull, we've got this major groove here that divides the brain into left and right hemispheres. So this is the cerebrum, again, this wrinkled portion of the brain. We have this long groove, and if it's a deep groove, we call it a fissure. So this is the longitudinal fissure that divides the brain into left and right hemispheres. And you've heard there's people that are left-brained and right-brained in that country song, right? Where most of us are a combination of both left and right brain activities, but we know that the left brain is a little bit more artistic and free-thinking, creative, and the right brain is a little bit more concrete, sequential, organized, mathematics, scientific. So if you tend toward one skill or the other, then it might be you're a little more left brain or a little more right brain, but most of us are a combination of both. So anyway, so that's the longitudinal fissure. And then if we look at the side here, there's a deep groove, which is called the lateral sulcus, or you could call it the lateral fissure. It's on the side. Lateral. So if you look at your sheep, or look at your brain model, it's on the side of the brain here. And it divides the lobes that we're going to talk about in a little bit. But this is the lateral sulcus, the lateral fissure. And then the central sulcus, not all of our sheep brains show this very well, but in this particular model here, I can see there's a blue and a red gyrus. And then in between there is the central sulcus. So it kind of sits on the brain like a crown. Remember the coronal suture on the skull? Well, the central sulcus lies in that same direction. It doesn't lie directly underneath the coronal suture, but it's still in that same direction, kind of sitting on the brain like a crown. So that's the central sulcus. So I can see that here. So that's the central sulcus. It divides the parietal from the frontal lobe. And we'll look at that here just a little bit. we'll get to the lobes, unless, let me see, do we have a lobe? Nope, we'll talk about that here then. Let's look at the lobes here. So, lobes of the brain. We have, looking at the first one listed, the front portion of your brain is the frontal lobe, which is like the forehead, which is here. So this is the frontal lobe. And then we have the parietal lobe, which is on across the top of the brain. So looking at this model here, this is the frontal lobe, and then the parietal lobe is on the top. So the back gyrus of the frontal lobe, that last gyrus, excuse me, this last gyrus of the frontal lobe is called the precentral gyrus. And why is it called that? Because this is the central sulcus, which divides the frontal and parietal lobe. So this is pre, which means it's in front of that central sulcus. So it's the precentral gyrus. That's swelling right in front. And this is important to know because when we get into particularly advanced A&P, we're going to talk about the function of that gyrus. A lot of activity is being sent to the brain there. And then in the back, behind the central sulcus, that gyrus, which is part of the parietal lobe, that's the postcentral gyrus. And this is what controls a lot of our motor activity. So it's like the central location that motor activity occurs. So damage to the postcentral gyrus is going to affect movement. Damage to the precentral gyrus is going to affect sensation coming in from the body. So these are two very important structures in the brain for function. This is called, another name for the precentral gyrus is the somatosensory cortex, and another name for the postcentral gyrus is the motor cortex. So again, we'll talk more about that a little bit in this class, but then more in advanced A&P. So parietal lobe, frontal lobe, on the side, temporal lobe down here, and on the back, you can guess, occipital lobe. Yep. And each of these lobes has a general function, and you do have to know functions of the brain, but it'll be given in a multiple choice format, so just recognize it, makes it a little easier, right? So looking at your lab objectives then, we'll go back to the generic ones. Frontal lobe is your thinking brain. So this is the part that you think with when you take your tests. You're really working that frontal lobe. And then the other, let's see, the parietal lobe is the orientation, recognition, sensory reception. So the parietal lobe has a lot to do with body movement and balance, orientation. And, oh, I think it looks like I had those backwards. Pre and post central gyrus. So this is the motor cortex, this is the somatosensory cortex. So motor control is here, somatosensory is here. But again, you're not gonna have to know details, it'll just be in a multiple choice format. The temporal lobe is where the nerves for hearing end and transmit their information. So for temporal lobe is hearing. And the occipital lobe is where vision is processed. So that optic nerve and optic tract ends in the occipital lobe, and that's where the processing of that light information turns into an image. So no general functions of each of these structures. And then we get to the diencephalon. We talked about it's that, central portion of the brain. So that circle that you're looking at right there, that purple, lighter purple circle, is the thalamus. And that's kind of like the secretary. Information comes up through the spinal cord, passes through the thalamus, and the thalamus sends it to different parts of the brain depending on what type of information that is. So we call it the sensory gateway. Information is coming in, thalamus will direct it to where it needs to go. And sometimes the thalamus will say, this is not important information, we're going to ignore that, and we're not going to make the body aware of this information coming in. For example, right now, do you have a shoe on your left foot? Can you feel your shoe on your left foot? Yeah. Were you thinking about your shoe on your left foot before I drew your attention to it? No, because your thalamus was like, not important that we be aware of that, right? But if something changes, like temperature changes and you start to get chilly, the thalamus will send that off and you'll be aware of that, right? So a lot of issues that people have with anxiety, overstimulation, anger control, impulsiveness, a lot of that has to do with the thalamus and sending information that others of us would not consider anxiety invoking. But if the thalamus operates differently, as it does in some people with, issues with anxiety and all that, it's sending that information to the alarm centers of the brain saying that this is stressful. So think of it as the sensory gateway. And then the hypothalamus is this region here. It's pretty boring to look at when you look at it in the sheep brain. It's not a significant, well-defined structure, but if you can find that circle of the thalamus, just go down below that, and this region here, this is the hypothalamus. Hypo means below. So the hypothalamus is the homeostatic controller of the body, which means it helps to control the endocrine system, and it has a lot to do with just general control of the autonomic nervous system, that involuntary part of our nervous system, just keeping everything in check. So that's the hypothalamus. So think of endocrine and autonomic control, really, really important. Body temperature, thirst, hunger, some of those survival skills and needs are controlled by the hypothalamus. And then hanging off the hypothalamus on a stalk is the pituitary gland, which has two lobes to it, the anterior and the posterior pituitary gland. So that's part of the endocrine system, so it's not part of the nervous system, but it's controlled by the nervous system. The hypothalamus controls the secretions of the pituitary gland, so you will see that. Some of your brain models may have it attached, that one sure does, but a lot of them don't. So if you don't see one hanging off of yours, then we're not going to have tested on that. But here's a good example of one. The pituitary gland is hanging off here on this little stalk. Here it looks on this model like a little peanut, right? So be aware that the pituitary gland is found there. Another structure we see, a small gland, part of the endocrine system, is the pineal gland, which comes off of the back of the brain, which is here. And the pineal gland, so you'll see it coming off the back of the thalamus, and we'll look at it in the sheep brain in a little bit. But the pineal gland, its job is to secrete melatonin, and that helps with our sleeping and waking up cycle. So you may or may not have heard that bright light, suppresses melatonin and keeps the brain awake and invigorated and not getting sleepy. So like if I turned the lights out in here and I was presenting the information on a dark screen, if you're tired, you might be like, oh, because that melatonin secretion goes up. So what does that tell you about a person who is really low on sleep? They've worked a long day. They haven't had much downtime, personal time to get caught up on Facebook. So they lay in bed with their screen in their face and they goof around on their phone with that bright screen. What is that going to do to melatonin secretion? Suppress it. It's going to suppress it, and they're not going to feel tired. So if you really need a good night's sleep, the best thing you can do, put your phone away, shut the curtains, create a dark environment for yourself. And even reading is okay if it's with a low light. So if you have a low light, where the light is going on the page, like if you have like a book lamp or something, and the light is going on the page, it's not coming from the page into your eyes, you'll find you'll get a lot tired more quickly than having a device that's throwing a light at you. And that's why they recommend kids stay off of video games. Like if you are a big video gamer, you should stay off of that, within an hour of bedtime, and allow that melatonin secretion to happen. And most people, as they age, the melatonin secretion goes down, and the elderly have a hard time sleeping. So if you ask most people in their 60s, 70s, and 80s, they're going to tell you that they don't sleep well. They get up early, and they usually have to take a little nap during the day, and that's okay. That's the way to cope. When we don't have good melatonin secretion, then you got to just do the best you can and take naps when you need to. Or some people supplement with melatonin. You can actually buy melatonin. You shouldn't take more than 3 milligrams, though, because it does affect the brain, and there's not a lot of research on melatonin supplementation, long-term effects, but 3 milligrams is what we see in in the healthcare environment that's prescribed. And you can buy it over the counter. You don't have to get a prescription. It's cheap. I take it sometimes in their little dissolvable pills. I just keep it next to my bed. If I'm having a hard time sleeping, I'll just dissolve one under my tongue, and pretty soon you just get that nice, comfortable, sleepy feeling. Okay, so moving on then, talked about one other piece to mention in the sagittal view. So we've got these two hemispheres, the left and right side of the brain, and there's that deep longitudinal fissure. Well, how does the brain communicate then between left and right sides? Well, we have this connecting layer here. So this connects the left to the right side of the brain, and it's called the corpus callosum. And you'll see that it is made up of, you know, a lighter tissue in your brain and in your sheep brain. So it's real easy to see that. It's the corpus callosum. It kind of looks like a flattened C in the brain. And that's how you can remember it. Corpus starting with the C and callosum starting with the C. Another name for it is commissural fibers. And commissural comes from the word commiserate, which means to communicate. So these are the way, these contain axons for communication between the left and right hemispheres. So that's the corpus callosum. So we talked about the thalamus, hypothalamus, pineal gland, pituitary gland, and then this just shows, again, the two lobes of the thalamus. If we turn the brain around and look at from a front view, we can see there's actually two lobes there. So there's a space between the thalamus. And then if we look at just below the thalamus, there's some other structures here. So in the front, we have what's called the midbrain. So the midbrain is made up of these two V-like structures in the front of the brain. The V-like structure is called the cerebral peduncles. So we're at objective number 7 right now. The midbrain is just below the thalamus, and it's made up of the cerebral peduncles, that V-shaped structure here. So there's two pieces to it. And then if I turn it around and look at the back side, I'm going to see 4 swellings on the back side, which are called the corpora quadrigemina. Corpora quadrigemina. Quad means 4, right? So those 4 swellings there that we see make up the corpora quadrigemina. So identifying these can be a little tricky, but if you know the parts of your brain stem, which is the midbrain here, the pons and the medulla, so find the swelling of the pons. The pons is always the biggest swelling on the brain. So if you look at your sheep brain here, can you see that swelling right there for the pons? Just above that, see the base, so you see the thalamus, which is that rounded structure, and you see the hypothalamus. It kind of looks like, if we go back up here, doesn't this kind of look like a, a seahorse? Here's the body of the seahorse. Here's the head of the seahorse, and then it has this kind of funny hair on the top. So the head and eye of the seahorse would be like your thalamus. The nose would be the hypothalamus, and the neck region would be the midbrain. So what's shown in green here is the brain stem. The 3 parts of the brain stem are the midbrain here, the pons, which is the swelling, and the medulla oblongata below that before it narrows down into spinal cord. So the midbrain, the front part, that if I turn it around and look at it straight on, it has that V-like structure. But this part right here is called the cerebral peduncles, and on the back are called the corpora quadrigemina. So this is part of the midbrain, just above the pons. So find the pons, and you'll find this right above it. Corpora quadrigemina. Cerebral peduncles in front, corpora quadrigemina in back. And then we have the pons, the big swelling, and then the medulla oblongata. So there's cerebral spinal fluid that is circulating around the middle of the brain here. And the spaces where it circulates we call ventricles. Just like the ventricles of the heart are chamber for blood, these are chambers for cerebral spinal fluid. So the parts that you need to know, we have the lateral ventricles, and the lateral ventricles are underneath the corpus callosum here, There's two of them on either side, so we're just looking at one side of the brain. So the lateral ventricles are underneath the brain, or underneath the corpus quadrigemina. Oh, I keep saying this wrong. Underneath the corpus callosum, we have the lateral ventricles. And then in between and around the thalamus is the third ventricle. So it's hard to see it in this side view diagram, but when we do the sheep brain dissection, and if you watch that video again after I do it here in class, you'll see I'll put my probe in the third ventricle. So you'll see these spaces better than looking at a flat diagram. But the third ventricle is around and between the two halves of the thalamus. So you'll find a little tissue in there that we're going to pull out, and this is called the choroid plexus. And it's made up of blood vessels and special cells that secrete cerebrospinal fluid. So it's a special little tissue that we'll find in that third ventricle. It's called the choroid plexus. Then we have this passageway, which is called the cerebral aqueduct. And the cerebral aqueduct takes that fluid that is formed and circulating in the lateral and third ventricles here, and it comes down the back of the brainstem, again, cerebral aqueduct, into this tiny little triangular space tucked into the cerebellum. That's the fourth ventricle. Just this tiny little triangular space right here is the fourth ventricle. Then that cerebrospinal fluid goes down and comes through the middle of the spinal cord, and that continues to bathe the outside and the middle of the spinal cord. And some of that also from the third ventricle comes up and goes underneath the arachnoid mater, right? Remember the subarachnoid space? So there's circulation that occurs there. But one thing to note about this cerebral aqueduct is it's a good landmark to find out where the corpora quadrigemina are separated from the cerebral peduncles. So the cerebral aqueduct divides those two structures. So here's the cerebral peduncles in the front and the swellings of the corpora quadrigemina in the back. So that cerebral aqueduct is a nice dividing line there. So let's look at, just refreshing our memory here again. Here's the space, part of the space for the third ventricle is between the halves of the thalamus and also around the thalamus. So the cerebral spinal fluid in that third ventricle is around the thalamus and between the two halves. And then talked about the parts of the brainstem. Looking at the ventricles again. So underneath the corpus callosum is the lateral ventricles. Then we have the third ventricle around the thalamus. Remember this choroid plexus? This is the special blood vessels and cells that make the cerebral spinal fluid. And then we have the cerebral aqueduct. We have the fourth ventricle here tucked in the cerebellum. And then you can see that the cerebral spinal fluid, circulates all over the brain and spinal cord. So it goes down the middle, it goes around the edges. Our brain is bathed in this fluid. When some people have too much cerebral spinal fluid during development, like as a child or infant, and those fontanelles are still there, remember where we don't have sutures over the brain yet, they get a really swollen head, and it's called hydrocephalus, because there's too much fluid, too much cerebral spinal fluid and not proper drainage. So they'll put a shunt, which is basically a tube to connect to the circulation system here that brings that fluid down into the abdominal cavity, and then the body just absorbs it. So that's a complication in some kids that don't have good circulation of this fluid around the brain. Then lastly, you have to know these coronal or frontal sections of the brain. So if you put the brain model together, it's got kind of a narrow end in the front, which is the frontal lobe, right? And then it widens in the middle and then it gets narrow in the back. So if I slice up this brain from front to back, kind of like a meatloaf, you know what a meatloaf looks like, right? If I slice it like you would slice a meatloaf, we would get frontal sections of the brain. And depending what slice I get, like at our house, some people like the end pieces, other people like the middle pieces. So depending where I slice, I'm going to pick up different parts of the brain. So in this picture, this is probably somewhere near the middle because I'm picking up those two lateral ventricles here, right? So there's two lateral ventricles, those are those spaces, and what do we call this deep line here that divides the two halves? So we're looking at, you know, a frontal view, so I cut it this way. This is the, no, not the central, because this is what divides the two, what is it? Longitudinal fissure. It runs the length of the brain. So it runs down the center here, right? Comes down here. So I'm slicing like this. So when I see that dividing line at the top, that's the longitudinal fissure. And then here's my two lateral ventricles. So right in the center here would be the thalamus. Let's go down and look at some other ones. Here we can really see it nicely. Here's my longitudinal fissure. Here's my lateral ventricles. Here's my third ventricle that would be around the thalamus. And then we have these other structures, these pie-shaped structures out here on the side. There's a better one here. Well, yeah, I guess, yeah. We'll look at this one. These pie-shaped structures are called the basal nuclei. And basal nuclei, the only reason we mention them is we want you to realize that these special structures in the brain are what controls motor activity. And the basal nuclei are what are damaged in people that have Parkinson's disease. The cells no longer respond well to dopamine, and as a result there's uncontrolled movements coming down the brain to the muscles, and they have troubles with tremors and a lot of movement. So we give them medicines to control that, but over time Parkinson's, those medicines work for about 10 years and then those medicines don't work very well anymore. Then they have to use other techniques like, you know, deep brain stimulation and all that. Like, what's his name, Michael J. Fox, who has Parkinson's. You know, he's got a lot of money, so he's getting the best treatments, and he's been able to go quite a while now with Parkinson's. But he's definitely affected by it. And it's the basal nuclei that are involved in that. So it's those pie-shaped structures, and they're best seen in this frontal view. Okay, so be sure that when you see this kind of frontal cut, you're thinking basal nuclei, ventricles, thalamus, just those structures you need to know. So if you look in bottom of page 15 in your blank lab packet, objective number 9, you know, you're just looking for those specific structures in a coronal or frontal section. So looking more here we can see the cerebellum. So this is a cut near the back of the brain that was made. So I can see that cerebellum there. So that's what that is. And I can see these lateral fissures here too, can't I? Or lateral sulcus on the side. This one's not quite as well seen, but I can see that here. And here I can see that longitudinal fissure, real obvious. So those are the different structures you need to know on the cerebellum. And again, the thalamus is just going to be in the center here, that center portion. So here's the thalamus, in the center of the brain. So we're going to look at the sheep brain next. I want to just identify, do a quick general overview, and then we'll get the sheep brain out, and I'll also have a video for you to watch. But so here's the sheep brain. Here's the longitudinal fissure. Here's the two halves, the two cerebral hemispheres, cerebral cortex. So again, gyrus are the swellings, sulcus are the grooves. Here's the cerebellum coming off the back of the brain. And at the bottom here, this narrow portion, this is what? Spinal cord. So cerebral hemispheres or cerebral cortex, cerebellum, spinal cord. If I separate, so if I pull this and separate it from the cerebellum, so I lift up the cerebral cortex, pull down the cerebellum, I see 4 little swellings here, 2 big swellings, 2 small swellings below. It almost looks like a baby's butt in there, doesn't it? So when you see the baby's butt, that's the midbrain, which is right below the thalamus, remember, but this is separating the cortex from the cerebellum, and this is the corporal quadrigemina. So that's that baby butt appearance that you're going to see when you separate those 2 structures. And then looking at the underside, so flipping the sheep brain over and looking at the bottom side, here's the olfactory bulb and the olfactory tract. So the bulb is the swelling, and the tract is this, you can see it's kind of this passage here, olfactory tract. And then we have this X-shaped structure in the middle here. So this, the top part of the X we said was optic nerve. Bottom of the X on either side was optic tract. And the crossover point in the middle was the optic chiasma. Yeah, the optic chiasma. And you don't need to know these two, so ignore that. Then we get down to the brainstem here. Here's the pons, so it's the swelling is a little, not quite as wide and large in the human brain, but there is a small swelling here, and that's the pons. So the pons is a lot more, you know, a lot shorter in the view here. So this is the pons, and then we have the medulla oblongata, which is this whole region here. And then again, when it gets to its narrowest point at the bottom, it's spinal cord. So medulla oblongata is kind of this long, oblong, that's where it gets its name, portion of the brainstem. So medulla oblongata, pons, and can you see the V-shaped structure here of the cerebral peduncles? That's what number 13 is pointing to. So you'll see it better when you have, you know, hands-on or watch the video. This swelling here and this swelling here makes up the cerebral peduncles. 10 and 11 you don't have to know. And then in a sagittal view, again, we see the cortex, we see the cerebellum, but you see this white, kind of lighter, flattened structure. What is this called here? Yeah, corpus callosum. And then the space underneath the corpus callosum, and just above it too, we could put our probe in there. That's the lateral ventricle. Here's the thalamus. See this perfectly round circle right here? That's part of the thalamus. So above the thalamus is the third ventricle, and this little light brown tissue tucked in there is the choroid plexus. So you've got the thalamus, third ventricle, choroid plexus, and this little gland coming off the back of the thalamus is the pineal gland. So you find the thalamus, and then the pituitary gland is here. It's been cut in cross-section, but this is the thalamus, so therefore this is the hypothalamus. This shiny space underneath the thalamus is the hypothalamus. And this is actually part of the optic nerve and optic tract here that's been cut. But this is the hypothalamus. Thalamus. Again, I can see the cerebral aqueduct here. So that means here's my peduncles of the midbrain. Here's the corpora quadrigemina of the midbrain divided by the cerebral aqueduct. Following this down, see where it tucks in this little space right here in the cerebellum? Do you remember what that's called? The fourth ventricle. Yep, yep. And then again going down to spinal cord. So those are the structures you need to know in a side view. You need to know your ventricles, so lateral, third, fourth, cerebral aqueduct, cerebral peduncles, corpora quadrigemina, there's the cheek of that baby butt, pineal gland, thalamus, the rounded portion, hypothalamus just below that. And then you have your pons, which is this swelling here. And then the medulla oblongata, which is much larger here in the sheep brain compared to the human brain. And then spinal cord down at the bottom. And then your cerebellum with the fourth ventricle. Okay, so I will have a video of the sheep brain dissection, and we'll do it here in class right now as well.